seems it's working now. Go ahead.
Okay, well, starting again. So I will present you my work, the work I achieved during my internship project, which was a study of the drought stress response among a maize panel. Well, first of all, what is it a challenge to address? So maize is one of the 3 most produced cereals in the world, along with wheat and rice, and so this is a cereal that's crucial to global food security. However, in the last years, the percentage of maize loss due to drought have increased. In this pie chart, we can see that the percentage of maize loss due to drought in Northeast China, one of the biggest exporters, this percentage have doubled between 1990 and 2017. So there is a clear need to tackle this issue, and a first step is to understand better the drought stress response of maize. So during my internship, I worked on data collected on a maize panel. So I worked in bioinformatics, so I had to work on data collected during experiments. So the experiment was on 254 maize hybrids run under normal or water stress conditions, and this experiment conducted over 4 years aimed to collect ecophysiological traits such as the biomass of the plants, the leaves surface area, or the sweating of the plant. Also, there were omics data collected each year, so the transcriptomic data, namely the RNA in the plant cell, and also the proteomic and metabolomic data, so the little molecules and the proteins that were expressed into the plant cell. So based on this, this design allowed me to investigate many different questions, mine being based on a master's project from a year ago to identify 2 groups in the hybrids, one having a significantly different response to water deficit. So here are my results. So basically, in the clustering, I performed a clustering independently on ecophysiologic, metabolomic, and proteomic data, and I could identify 2 groups, one being significantly more responsive to water stress. So here we can see that the smallest group, which is cluster 1, have a significantly higher average for every ecophysiological trait, and also this small group shows more variability in its response than the bigger one. This 2-group structure can be also identified by a clustering based on proteomic and metabolomic data, which is very interesting because it shows a common structure on the 3 data sets. This common structure can be a proof of the relevance, biologically speaking, of the clustering, and it also shows that this 2-group structure can be seen also at molecular scale, which is very interesting when it comes to identifying the molecules responsible of the maize drought stress response. So to conclude, these works have many perspectives. The first one is a work to highlight the more important molecules of the process, which is the first step towards understanding and identifying the key mechanisms of water stress response for maize. And also, this 2-group structure can be integrated in the genetic association study the PhD student of the team is currently performing. So it's really an interesting work. Thank you for your attention.
Thanks. So the next speaker is Jael. We'll try to find. Oh. Now what happened? Seems as when I go to this, it doesn't, okay. Okay, okay, yeah, so this is the pointer, and here if you have difficulties, maybe with it.
Thank you.
So the floor is yours.
Thank you. Hi, and thank you for attending this session. My name is Yael Aiden, and I am a first-year student at the ENS Paris-Saclay, which is equivalent to a bachelor's degree in biology. The internship that I will be talking about is part of my bachelor's degree here at the ENS, and it will actually take place next week. So I'm not yet in there, and it will be at the Weizmann Institute of Science in Israel, and I'm really looking forward to it. Yes. Now let's talk cellular biology and signaling pathways. This is a human cell. It is the very basic building block of all of our body. You can see that it's got some very interesting features, including a very specific compartment that is called the nucleus, right here. The nucleus is enclosed in something that's called the nuclear envelope, and it is what contains the DNA. Now, one very interesting feature about human cells is that they are able to react to their environment, meaning that they can perceive somehow extracellular factors such as molecules that will be present in the extracellular media, and then the cell can perceive these factors and respond to them appropriately. How that works, in a very simplified way, is that the cell actually bears receptors that have an extracellular portion right here that can bind to the factors, and when they are activated by these factors, they trigger something that's called a transduction cascade, which is a series of reactions that activate and activate and activate a lot of factors that eventually activate one last factor that's called the transcription factor. And what's very interesting about transcription factors, thank you, is that they then need to enter the nucleus in order to bind the DNA and then regulate a whole lot of genes that will then confer the cell response, the response to the first signal, remember? Yep. So this response actually depends on a whole lot of genetic networks, and it really requires this interaction of the DNA with the transcription factors. This whole process is called a signaling pathway, and it depends on a few major parameters that we can identify using this schematic. First of all, there's the signal intensity, so the concentration of the extracellular factors, for example. Of course, there's the receptors quantity. The more receptors a cell has, the more likely it is to be activated and to perceive the signal and then respond to it. There's also the transduction efficiency. So this little purple arrow that I have not detailed at all, but it's actually a very, very complex series of mechanisms, and that plays a very important role in the signaling pathway. And then there is this last step, that is the entry into the nucleus, and that I will talk about today. The question that arises is how the transcription factors is actually able to enter the nucleus and cross the nuclear envelope. And that is allowed by the nuclear pore complex, which is a protein complex that is found within the nuclear envelope, and it actually crosses the envelope, which actually gives a certain continuity between the cytoplasm and the nucleus. So it's actually very much like a little channel that allows transcription factors to shuttle through from the cytoplasm to the nucleus and actually bind with the DNA. What's very important about the NPC is that it only allows for certain types of proteins to shuttle through. So it is actually a mean of selective transport. Other proteins that transcription factors are not guaranteed shuttling through. And again, what I would like to draw your attention to is NAP93, which is a protein that's part of the inner ring of the NPC. Now, why is it relevant, and how is it going to take up my whole summer? Well, first of all, it has been identified by bioinformatics studies as overexpressed in patients with metastatic breast cancer. these first studies have actually led to more trials using mainly mice models as well as in vitro cell manipulations, which have proven that NUP93 overexpression, so too much NUP93 in a cell, promotes tumor growth and metastasis. And last but not least, biochemical assays that were motivated by these previous findings have shown that NUP93 overexpression, again, promotes excessive translocation of cancer-related transcription factors within a cell. So to piece the puzzle back together, we have the first diagram, and we remember that the transcription factor has to shuttle through the NPC to reach the nucleus. But if there's too much NUP93, we can hypothesize that this causes an enhanced nuclear entry of the transcription factors, which in turn de-equilibrates the signaling pathway, and eventually leads to metastasis instead of a regular cell response to a regular signal. Thank you for your attention. My internship will actually focus on the molecular mechanisms regulating this whole pathway. Thank you.
Thank you, Brian. So our next speaker is Theo. Check. Yes, it's working. So the middle, the pointer, and okay. Go ahead.
Hello everyone, I'm going to present you my research project using neurofilm electronics in order to build a really low power consumption neural network. So first of all, what is neurofilm electronics? It's actually combine the brain cell that you all know about on the electronic paradigm with silicon chip on micro dimensional space. You mix together, you don't get terminator yet, but in the state of the art, you have a really low implementation of few neurons, or only one in this case. So what is actually how function one electronic neuron? So the researcher try to get the as the biological implementation step by step. So if we consider the travel of the information going from the top to the bottom of the human neuron, first of all, there are displacement of ion in the dendrite. So this has the equivalent in the electrical circuit with input current. Then, as the information go through the membrane voltage, here you have voltage and a capacitor at the back of the circuit. This will create an oscillation. As in your brain cell, you have around 10 Hz oscillation. Here you have about 40 kilohertz. Not to be smarter, but because you save space working with smaller capacitor and higher spike frequency. Then, as you have a little time of reflection, you have a propagation delay between the nucleus of the cell and the axon. So this is emulated through RC filter implementation going characteristic time delay. Finally, as in your brain you don't have only one neuron, you want to connect together with other neuron by layer, and so you want to copy paste the information. This is occurring in the electronic version through a current mirror that copy the current flowing with a different gain. So just to be more precise, why you want to use electronic neuromorphic? Because you really save a lot of power, moreover in a context of ecological transition, to give you the idea, a classical implementation of such network will use a graphical card, and a graphical card has the power consumption of 175 million of tiny electronic neurons. So this is huge, and you have really an advantage to work with neurofilament electronics. And in this planning, the main component is the activation function, which describes how your neuron behave, and the weight, which describes how two neurons are strongly connected together or not. So why is a research project? Because actually it's really hard to build those network with those tiny neurons. If we consider one neuron, Bob, and you want to connect with other on doing some smart stuff, you may want to use a classical analog circuit simulator, but it's very slow. So the more convenient way to do that is try to copy-paste existing network, digital network, and try to implement them analogically. So if you want to connect Bob with another neuron Alice, you need to put a connection part, the dendrite, that propagate the information. But if you connect another dendrite, there are influence that would not have in a classical digital network. So this is one problem. If your gain are too small or too big, you have again other issue to design. And each dendrite that you add want to retro-propagate his behavior in the neuron that makes the sizing again difficult. If you want to inhibit your neuron to not get the network crazy, over-excitated, you can do inhibition, but again, it's also a problem of non-linearity. But a good point with Bob is that he's not able to really look correctly at the front layer of him, so you can do everything you want. If there are the same current, there will be the same response, so it's very a good point for sighting. And finally, what is the main challenge of implementing low consumption neural network? It's designing them with the strange behavior of Bob. Thank you for your attention.
Thanks. Just give me a couple of minutes because I need to download another presentation. Okay.
Dear viewers, dear spectators.
So the next talk is a video of Baptiste Piguet. Well,
dear viewers, dear spectators, good afternoon. As you may know, aging is accompanied with an increase in the rate of several diseases, including cardiovascular diseases, cancers, or type 2 diabetes. This is why the aging of population is a major global concern, and the understanding of causes of aging an important challenge, because it could lead the way to treatments that could improve the health of aging people. Now, aging is also a very observed phenomenon that exists in many organisms, even in the budding yeast Saccharomyces that is used to make beer and bread. DNA damage is thought to be an important cause of aging since several decades, and at the beginning we rather thought that DNA damage caused aging by generating DNA mutations that would accumulate throughout the life of cells, which would cause the frailty observed in the age of the organisms and eventually their death. Now, nowadays we think that this is rather unlikely because firstly, human individuals who accumulate many more mutations than healthy individuals do not witness an accelerated aging, while yeast, right before they die of old age, usually accumulate 1 or 2 mutations on. But on the other hand, we also have a great body of evidence suggesting a link between DNA damage and aging, including the fact that proteroid syndromes, diseases causing people to accelerate and to age in an accelerated way, well, these proteroid syndromes are often due to dysfunctions of DNA repair genes. As I mentioned before, DNA damage can cause mutations, but not only. It is not the only consequence it can have. It can also block or stall replication forks when they are copying the genome, and this can of course make replication harder and activates detrimental signaling pathways. So my project in the lab of John Housley had the aim of study of determining whether replication stress existed in each cells, and further to study what is the importance of this replication stress if it exists. So to answer these questions, we modified the genome of yeasts. We modified a gene called MRC1, which is activated by blocked replication forks, and in turn activates the replication checkpoints, which is, to be simple, a response that allows the good management of blocked replication forks and allows them to restart faster. And we replaced these genes in the yeast with another version called MRC1AQ, which cannot be activated by stalled replication forks. So we basically aged normal yeast with a normal MRC1 or yeast with MRC1AQ, and then use the technique that is called the trial-seq that can reveal where replication forks are blocked inside the genome. And so, if replication stress existed in aged yeasts, we would expect yeasts with MRC1AQ either to have problems earlier, either to have more problems at an equivalent age. So when we looked at our trial-seq results for with normal yeasts, with a normal MRC1, we saw that replication stress increased inside regions that are not covered by histones, and therefore more vulnerable. And we also saw a profound disruption of the replication patterns of this very aged normal yeasts. And both of these phenomena appeared to occur earlier in yeasts with MRC1AQ. So we think that this increase in replication stress in histone-free region could be due to the fact that here DNA is innate and therefore more susceptible to oxidative damage. That is, we have evidence which proves that in the literature. But for the disruption of replication patterns, we are nowadays unable to explain that. We don't really have a convincing model. So, of course, we have many more questions that are left to be answered. We would like to test our model. We would like to understand what causes the disruption of replication in aged cells, and of course, know how really important is the replication stress in aging. So finally, I would like to thank all the people who helped me during my project in this year in the Abraham Institute.
So our next speaker is Nina Granger. Well, let's see if it works. Yes, it's working.
Thank you.
The middle is the pointer.
Hi, everyone. My name is Nina Granger. I'm part of the English department here at ENS Paris-Saclay, and I'm going to try and present quickly my master thesis project. My master thesis project that I'm going to defend at the end of this month. It's entitled Writing in the Fabled Damned of Nations, America, Poetic Ethos and Decay, and Allen Ginsberg's reading of Walt Whitman. So, of course, I'm not going to summarize the works of Ginsberg and Whitman. It would be too difficult. They are two seminal American poets, and I'm going to try and present to you the basis of my work and how I approached this filiation. So what prompted this work, this study of Whitman and Ginsberg, is a single line in a poem. Sounds crazy, right? But it's this line, with thoughts, I have a view tonight, Walt Whitman. It's a very famous line from a poem entitled The Supermarket in California from Ginsberg. And as a reader, I feel like this sentence was directed to us readers. What thoughts does Ginsberg have about Whitman, a poet who wrote 100 years before him in a similar context of crisis? Whitman after the Civil War and Ginsberg during the Vietnam War. So I tried to study this filiation through the idea of the fable damned of nations. The fable damned is a biblical phrase that Ginsberg borrows from Whitman, and he explains in his 1974 speech that Whitman had prophesied America's decay. And he uses this phrase that is from Whitman's Democratic Vistas. So in this speech, Ginsberg sees Whitman as a prophet. And this brought me to a very common thread of thought, that is the idea of the poet in America. So in American studies, the poet-prophet is a notion that is widely studied. And I wondered in my thesis, how does one write in a country that is falling, if we follow Ginsberg's appreciation that America is indeed falling in the wake of the Vietnam War? What if the poet himself is America, as Ezra Pound pointed out about Whitman? What happens to the poet then when America falls, if he himself is America? Another seminal text is Emerson's The Poet, in which he explains the status of the American poet that is both aesthetic and ethical. So this brings me to my last notion, that is maybe the most controversial or the most interesting, both in research and my thesis, that is the notion of ethos, and more specifically of poetic ethos. Ethos is a notion that was developed by Aristotle in the Rhetoric. So the three modes a narrator can communicate to his audience and stand as a powerful figure are ethos, logos, and pathos. Ethos is linked to the way the orator is perceived by the audience and the intrinsic moral qualities the audience perceives in the orator. In poetry, this notion is really interesting because it's been studied through its manifestations in the text. Dominique Mangoneau explains that there is a discursive ethos. That means that ethos is not outside the text, it's within the text. The poet, or the writer in general, acts and behaves in a way textually that makes his readers, or their readers, trust him. So this idea is a socio-poetic idea, the idea that the poet is not only a writer, he is also a textual figure that appears through the text. So maybe some of the questions that I'm going to raise in this thesis are what is the poet's role in times of crisis? How is this role displayed textually in Ginsberg's writing of Whitman through a method of close reading? Thank you for listening.
Thanks, Nina. So the next speaker is Eva Dumenil.
So, hello everyone. So I'm here to present you a project I've been conducted as part of an agroparitech internship conducted by Fabrice Requier and Clémence Riva. So I will ask myself, how important are pollinators for food security? So as you all have heard about, we all know that pollinators are declining, and particularly bees, and it impacts our agriculture. So the FAO reported that 1/3 of our bees and butterflies are declining. They also say that 10% of them are endangered. Another study made in the US show that honeybees are declining since around the 1950s, 1960s, so when we had some intensive farming and industrialization. But why are they declining? So first of all, climate change, but that's not the only reason. There's also pesticides and herbicides, which are used not on a particular insect, but on all the insects. Also, the lack of flowers in open field landscapes, and some pests, such as the varroa, are destroying our insects. But why is pollination so important? So in order to give fruits, the flower has to be pollinated. It means that the pollen has to be transported from stamen to pistil, and then it gives the fruits. So we've been asking 3 questions. Is there a pollination deficit? So does pollinator decline reduce the crop yields? Is apple production dependent on the presence of insects? And also, do pollination services improve apple quality? So we will try to find the intermediary crop basket between the crop we can produce without pollinators, so that's just 4 of them, and here the crop we can produce if there's an optimum presence of pollinators. So, and we will focus on the apple. So in order to answer this question, we've been conducted experiments in 2 regions of France, so the Ile-de-France and the Vaucluse. In each region we had 5 apple orchards, and in each 5 trees. And on the trees we had 3 treatments. So the first one is to exclude all pollinators of the flower through the use of a net. Then you have the other one, it's just open, so the pollinators can come. And here it's open, and also we added a human pollination. And then we made 3 measurements. The first one is the fructification success. So we counted the number of flowers and then of fruits. It gives us the yield and then the loss of flowers. And then the seed set is the number of seeds per apple and per treatment. And the apple weight is also per treatment. And we've got some results. So those results are based on a linear regression of all the 3 explanatory variables as a function of the treatment. And as you can see on the top of the graph, there's letter. So if that's a different letter, then it highlights different statistical differences between the treatments. So as you can see here, there's a different letter for the exclude one. So the fructification success is less important than when it's just open. You have the same results for the seed number. And finally, as you can see on the last, so the apple weights, the letters are the same for the 3 treatments, so there's no statistical differences between all the treatments. So to conclude, we can see that there's no pollination deficit, so no need of human supplementations in order to produce our apple. And we also have seen that the quality of the apple, so here the apple's weight, isn't based on pollination, but we need our pollinators in order to have a yield which is more important, because apples are dependent on insects, and that's why we need to protect them. Thank you.
Thanks. So the next is Matthew. Okay, in the middle of the pointer.
Hello everyone, my name is Mathieu Lordez, and my presentation will be on the internship of 5 months that I did in PPSM this year for my BioPro personalized project. The goal of this project was to conduct the pluridisciplinary study of photoswitchable glycomicrocycles for metallic ion complexation. This project started a year ago when this kind of molecule was synthesized by a member of the team. For this presentation, I will only focus on this one called E21 beta. It has been designed to have 3 main characteristics. First, the upper part of the molecule is a group called an azobenzene. When an azobenzene absorbs a photon, the central bond will change its configuration, and it will change a lot the electronic properties of the molecule. The second important group are the sugar group at the bottom. These groups are chiral, but they are transparent in visible light. However, there is a chirality transfer from the sugar group to the azobenzene, which means that the study of this molecule in circular dichroism is possible. And circular dichroism is a method used to study chiral molecules like DNA or proteins. The last important group is the macrocycle as a whole, because this macrocycle on its heteroatoms like nitrogen and oxygen form a stable cavity for metal ions to go in and complexate with this molecule. This molecule has been designed to combine optical and complexing characteristics because of the potential application in biology. Whether it's for depollution by detecting and removing heavy metal ions from water, or in biological processes to regulate and monitor these processes. But different methods are needed to properly characterize this type of complex molecule. UV visible absorption and circular dichroism are used together here because of the complementary information they give. When this molecule occurs a simple photo-switching reaction, the electronic properties of the molecule will be greatly changed, and that will change the UV visible absorption spectrum. But it will also change the whole conformation of the molecule, and these changes are observed, are tracked in circular dichroism by change in LECT. When a complexation reaction occurs between this molecule and a metal ion like copper 2 plus ion, the copper 2 plus ion will slip in the macrocycle cavity of the molecule, and it will interact with the azobenzene. This will lead to a great change in the signal in UV visible absorption, shifting the whole molecule to a red color, and it will also change the LECT and the conformation of the molecule as observed in circular dichroism. But these techniques are not enough to properly characterize this molecule and confirm that the phenomenon in action here is really metal complexation. The confirmation method that we used was mass spectrometry, because in mass spectrometry we are able to see the signal of the macrocycle that is not complex, and the one that is complex with a copper 2 plus ion, which has a higher molecular mass, and a 2 plus ion. With this method, it was also possible to confirm complexation with metal ions that does not change the UV visible spectrum or the circular dichroism signal. The study of this kind of molecule is still in an early state due to their recent synthesis. However, other methods can be used to completely characterize this kind of complex molecule. For example, calculations have been tested to see the optimal conformation of the molecule in theory, as you can see here on this graphical representation of an energy minimization of this molecule. And other methods like NMR or EPR can be used to characterize the interaction between metal ions and the macrocycle completely. Thank you for your attention.
Yes, so the next talk is done by a team.
Yep.
That's great. So, I think he's... C'est comment pour... Normalement, c'est... Voilà. C'est le pointeur.
So, hello everyone. So we're here today to present you our supervised research work that we carried out at the FAST laboratory in University Paris-Saclay. And it is entitled Added Mass Force from Fluids to Dry Granular Matter.
So, first you may ask, but what is granular matter? So, granular medium is a system made of a lot of small solid particles that, taken together, behave just like a fluid. If I say coffee grains, scree, or even sand, you can see what I mean. It can flow like a fluid, and it has also no proper shape. And those fluids are really interesting to study. Sometimes they can jam and stop flowing. They can also crystallize and then change their mechanical properties. And the granular matter science has many applications from civil engineering to space exploration, and it's also used to better understand the snow and rock avalanches.
So, our main idea during the study was to take a textbook case in fluid mechanics, which is a sphere immersed in a Newtonian fluid. We applied a certain speed u to that sphere, and we measured the force applied to it. And we tried to compare the well-known fluid mechanics results with the results in dry granular matter, and wonder how can we generalize the fluid mechanics results to granular matter. So, to explain it quickly, in fluids, we didn't show the questions just to show off. We showed them because in fluids, we have theoretical results. And we are lucky from that because it is much more simpler than purely experimental or simulated results. So for a sphere, you've got first what we call the steady drag force, which is a force that applies on the sphere at a constant velocity, and it is a force depending on velocity. And for unsteady movement, when you accelerate your sphere, you've got force which is linearly depending on acceleration. That adds to the steady drag force. In dry granular matter, it is much more complicated because it is a non-Newtonian fluid, you don't have any theoretical results. You have to rely simply on simulations and experimentations. Our supervisors performed a few simulations. They first demonstrated numerically that the steady drag force was constant at low velocities and quadratic in velocity for high speeds. And they also showed that we had an added mass effect in dry granular matter, which meant that they measured a force that was linearly depending on acceleration.
So our aim during our study was to reproduce the simulations, but experimentally. So here you can see a screenshot from the simulation where a disk is moving in the two-dimensional granular medium. And we made the same thing, but in the real world. So here is a granular media composed of small plastic beads in which we immersed a sphere. This sphere is moved thanks to a huge electrical cylinder with high performances. It can accelerate up to 1 G. And between the sphere and the cylinder, we got a strength sensor which will be measuring the forces over the sphere during its movement. So here is what we got after one measurement. The x-axis is the position. So if you look at the speed plot, you can see that we imposed first a small speed plateau in order to establish a force chain in the matter, and then we made the sphere accelerate until it reached a nice speed plateau, and we will be looking what is happening between those two phases. So here is the force plot. You can see that first we jump to a small force plateau, and then we reach a high force plateau. And the difference between the two plateaux is added mass force.
So, to present our results quickly, we were able to demonstrate that at low velocities, we had clearly a constant steady drag force, which was in accordance with the simulations. And we also showed the existence of a force depending linearly on acceleration, which we can interpret as added mass effect. So this is quite encouraging for further studies, especially about characterizing what we call the added mass coefficient, which is another parameter. And that's all. Thank you very much.
Thank you. So the next one. Okay, so this is the pointer, and this is your. Can you go close to the microphone?
So, hello everyone. The work I'm going to present today was realized as part of my master's degree here at the ENS Paris-Saclay and at the Université Paris-Cité. My supervisor was Emmanuel Ferran, professor in phonetics at the UPC. So my aim was to study the shift in pronunciation that happened amongst Scottish rock bands towards the 90s, when they started to sing in their own accents rather than adopting an English or American one as they did before. And so my work consisted in two parts. First, an acoustic analysis of 4 bands, and then a discussion around existing sociolinguistic theories on style shifting, which is what we call shifting speaking style. So for the acoustic analysis, I focused on the discography of 4 bands. The research hypothesis was that the younger bands, Arab Strap and the Twilight Sad, would have a more Scottish accent than Telemetry and Primary Scream, but that these two would have would show a possible evolution, moving away from an American accent towards a more Scottish one. And so I studied 2 phonological variables, which mean that they are pronounced differently depending on one's accent. And the first one was the face, the face and goat vowels. When I say face and goat, I mean A and O. Most Scottish accents realize them as monophthong, meaning one single sound in the vowel instead of 2. So instead of A, you have this, and instead of O, goat, you have goat. And the second one was side and tide, as written on the board. A bit more complicated because this difference has to do with morphology. But basically, if you only have one unit, as in tide, the waves, it's typically pronounced tide. And if you have a morphological boundary afterwards, so in this instance, you have the verb tie, and then you have the past suffix d, so a morphological boundary, and that is pronounced tide. So I focused on that. What do we study in phonetics when we study vowels? So first of all, we use a software called Pratt, and then we look at formants. Formants are these dark shade bands, darker shades of bands you see on the spectrograms. They correspond basically to high concentrations of energy at certain frequencies, and we measure these frequencies. And so to study whether a vowel is monophthong or a diphthong, you look at whether there is a change in frequency, in frequencies, in formant values. Here you have a change, rising. Here you have no change, basically. And then the second one was a bit more complicated, but I used a method called discrete cosine transform, which basically calculated a series of 30 coefficients to allow me to study the trajectory of the formants. And here are the curves reconstructed from those coefficients. And so that allowed me to see the differences between in pronunciation. Yeah. My results, or the measurements I got, were analyzed using Bayesian statistics. And that showed me that Arab Strap and the Twilight Sad did have a more Scottish accent, as expected. But sadly, I did not see any noticeable evolution in Telemetry and Primary Scream's discographies. So I could not actually verify my hypothesis. I would need a much, much bigger corpus with many bands and conduct a statistical analysis on a much larger scale, which was not possible this year for me. Then I analyzed the results based on sociolinguistic theories on style shifting. So I looked at Trudgell's notion of admiration, so basically adopting an American accent because American bands have dominated the genre of rap music. That is that. Bell's idea of outgroup and in-group referee design means that I might adopt a different accent if I want to project a different identity. That is outgroup, different from my in-group. And if I want to identify or project the identity of my own in-group, that is in-group referee design. And then finally, we had to consider, I had to consider the importance of lyrics. So the mode of discourse is what it's called in linguistic, and as well as the physical constraints of singing and what is more singable, what is more sonorous. Diphthongs are more sonorous because you don't close to a close vowel. And so that's it for me today. If you want to discuss it further, do come and discuss with me afterwards. Thank you.
Thanks. So the next talk is a video. from Victor Lopez de Sousa.
Hi everyone, my name is Victor and I'll present you in this short video some concepts in a theorem in pure maths. It might start seeming abstract, but the important thing are the drawings. Let's go! Groups. Groups are sets with an operation that follow three simple rules. But, well, this may not mean a lot to you. The important thing to know about the definition of groups is that they are a collection of things that can be put together to form other things that are also in the group. And also, which is made the most important thing, groups possess, in some sense, a symmetric structure. That is, they have a neutral element, and every single element has an opposite correspondent as a reflection. The classical example is the integers, that are the numbers that you already know. They are 0, 1, -1, 2, -2, 3, -3, and so on. In resume, the integers are all possible values that one can form by summing 1 and -1 an arbitrary number of times. 0 is 1 plus -1. 4 is 1 plus 1 plus 1 plus 1. -4 is -1 plus -1 plus -1 plus -1, and so on. The only thing we need to know, then, to write any integer to define this whole group is what is 1, what is -1, and how to sum. Then, we can say that this group is generated by the set of 1 and -1. As the set is finite, or more precisely, as we can find a finite generating set to this group, one can call this group a finitely generated group. It's about these groups, finitely generated groups, that we'll talk about the rest of the video. And well, I can understand this definition may sound kind of arbitrary and abstract, but for now on, it will be mostly about drawings. I swear. So let's draw this group. That may be the only group we know, but anyway. A very natural way to do this would be to put a point to every element of our set, and to link them with the operations induced by our generating set. That is, link every element to the ones we can find by adding 1 or -1. These drawings are we call Cayley graphs of a group. We'll see some more examples in a bit, but for now, it's very interesting to see the geometrical nature of this figure. And, well, what do I mean by this? Basically, considering the Cayley graph, we can talk about the neighborhood of a point, the distance between 2 points, the size and shape of any subset I want. And because of this, we can talk about properties that didn't make sense to the group when we analyzed it before from a pure algebraic point of view. And that's exactly what we'll do by studying the number of ends of a group, which is a very classical thing to do in geometric group theory. Ends. Intuitively, ends are the number of ways in which we can go to infinity without leaving the graph. What does it mean? Well, imagine I paint one vertex of the graph. It corresponds to an element of the group. I would still have two disconnected white things that are not painted. If I now paint the edges and vertices that are immediate neighbors to this first point, I still have two disconnected no painted subgraphs. And if I painted all the vertices and edges that are neighbors to something that I already painted, I still have two disconnected, not painted things. And it doesn't matter how many times I do this process, I'll still have two disconnected things. So I'll say I have two ends. This graph has two ends, and this group has two ends. Let's dive in some other examples then. Starting by Z2, which is basically the integers and the integers again. An element of Z2 is a pair of two elements of Z, which is the integers. Its Cayley graph is like we show in the screen. It's easy to see that repeating the same process that we did last time will always result in one non-painted connected element, so we'll have one edge. Another example are finite groups, like these that will appear on the screen. They come from a construction that we call modular arithmetic, and for a number n, we call them Z mod n. One can think of them as the first n natural numbers. The group structure is given by the unitary sum, with the special rule of the last element plus 1 equalizing the first. This provides the cyclical nature of the Cayley graphs. We can see on the screen, respectively, the ones corresponding to Z mod 3, Z mod 5, and Z mod 7. It's not hard to see that finite groups have zero ends, as when we start painting, we suddenly paint it all. The last group I want to introduce you is a bit different from the others, as its elements are not usually understood as numbers or pairs of numbers or anything like that, but words. And what do I want to say by words? Well, imagine a very simple alphabet with A and B. And, well, we're talking about the group, so we must have symmetry. Then we must consider minus A and minus B. This group is usually called free group, and it is the collection of all possible words, considering the word that is no other empty word, and adding some number of A, B, minus A, and minus B. Our generating set is, then, the four possible letters, and the operation of this group is the act of adding a letter in the word. And how we draw is, well, we start from the empty word and start adding letters after this, as in the image. The result will be this very cool graph with a fractal nature. And what's the number of ends? Well, you can see, as we start our process of painting, the number of non-painted disjointed things will just go up and up and up and up. Then, we'll say this group have an infinite amount of ends. Now, it's very natural to you to wonder what other groups exist. How can I draw them? How many ends do they have? And I can assure you, there are a lot of things out there. If you can think about a finite generating set and operation, you can construct a group, a finitely generated group. You can then draw their Cayley graph, as we did, and calculate the number of ends. But I can assure you, and this is surprising, maybe the highest point of the video, the final result won't be new. I showed you all the possible examples of number of ends. It's crazy, doesn't it? We have our theorem, and I can state it right now and you understand. Every finitely generated group has 0, 1, 2, or infinitely many ends. This is not trivial, this sounds arbitrary, but it is what it is. There's no finitely generated group with exactly 3 ends or 4 ends or whatever. Well, to show rigorously why is not trivial, but you can always try to read my paper, which I prove this. I guess that was it. Thank you very much for your time, and see you next. Bye-bye!
Is it Maxime Coca is here? I don't think so, so we will go to the next. Is Jennifer Calderon? Yeah. So Jennifer 37. Okay, Jennifer, so this is the pointer and you just displace.
So, good afternoon. My name is Jennifer Calderon, and it's a pleasure for me to be here today to present to you a bit of what has been my M2 internship, which I'm working right now. I'm probing the fluorescence of conjugated polymers in the vicinity of plasmonic surfaces here at PPSM, just in the building right next, under the supervision of Professor Fabien Miomond and Dr. Vitor Brasilens. So, in this presentation, I will be addressing a lot what are electrofluorochromic conjugated polymers, which sounds a bit long and complicated, so let's break it down a bit in some interesting concepts. First, a conjugated polymer is characterized because of its ability to conduct electricity, which might be counterintuitive at first, because when we think of polymers, we think of isolators like plastics. Then the effect of electrofluorochromism is the ability to switch on and off fluorescence with an electric impulse. Then we want to stimulate these kind of systems using a plasmonic effect, which is shown in metallic nanoparticles, where we have electrons very close to the surface and that can interact very easily with light, and they will generate charges that can interact with our system. These systems are very interesting for applications in molecular electronics, which is a field that is studied in efforts to further, for example, miniaturize more our electronics. And these polymers are very interesting because they are conductive, but they can also have an optical response to electric stimuli. So they can be used, for example, in displays or in sensors. What we want to do is to implement also a plasmonic control through metallic nanoparticles plus the electric response, so we can have a multimodal control of this optical response. For this, we divided the project in 2 parts. The first one, we want to just understand how is the fluorescence related to the oxidation state by electric stimuli of our system. Then we will use this system to construct a molecular junction, which is just to put our polymer in between 2 electrodes. And we will use this to understand the effect of current and also the plasmonic excitation in our system. We started by producing the polymer, so we can do this by 2 methods. The first one is by applying a voltage. In here we can produce this kind of film by oxidating our molecule. Then we can also do it by using light, by exciting it with a laser. And we can actually do more complex figures. These are dots, but you can kind of write as figure as you want. The interesting thing about having 2 different methods is that let us do and design different kind of experiments, as I will show in a minute. We want to first understand this switching of color and fluorescence while we apply the voltage. And for this, it's essential that we can observe simultaneously the electrochemical and the optical response. So for this, we use this setup designed by the group, where we couple an electrochemical cell and a fluorescence microscope. We put our sample here, and we apply a voltage. And with the fluorescence microscope, we excite it using light, and when we will measure is the light emitted by our sample, and this is how we can see when the fluorescence is turned off. And now that we understand better our system, we can use it to build more fun things. First, for example, this kind of a simple molecular junction, we use it by using a laser, and we produce it in between the two gold electrodes. And this junction is fluorescent when there is no passing current, and then when we pass a current through it, we get a decrease in the fluorescence. This can be very interesting because then we can clearly observe when it has certain level of current already been surpassed in our junction. Then we can use the other type of polymerization by applying a voltage to build other type of molecular junction. In this case, our electrodes will be an ultramicro platinum electrode and a plasmonic surface, which is just this surface which is conductive and gold nanoparticles on top of it. Then with light, we will generate a plasmonic excitation, and we will generate electrons on top of the gold nanoparticle, which will interact with our polymer here. And this will generate changes in the conductivity of it, and we will be able to detect this by fluorescence. And what is very nice about this is that since we have electrodes here, we can also simultaneously apply an electric stimuli. And this is, at the end, the final take of our project, where we can build and understand the system and get more fundamental understanding of how this system with electrofluorochromic polymer on top of a plasmonic substrate can be modulated both by electrochemistry and by light with the plasmonics. And this is very important because in the future then we can use this to develop further molecular electronic devices, for example, with optical response. Thank you very much.
Okay, so our next speaker is Sadia Akhter. 38. Yes, checking. Okay, so pointer and these to move the slides. Okay, go ahead. Can you try to be close to the microphone? Works?
So, hello everyone, I am Sadia, a Master 2 student of University Paris-Saclay, and currently I am doing my Master 2 internship in the IPS2 on the project entitled Role of Poly-ADP Ribose Polymerases in Plant Adaptation to Highlight, supervised by Berto Gakir and Livia Marangino. So, the story starts with NAD, which is an important molecule in plants because it's involved in more than 3,000 enzymatic reactions. And under abiotic stress conditions, for example, high light stress, this NAD is consumed by this PARP protein to produce multiple ADP riboses on the proteins associated to DNA. And it leads to resistance in the abiotic stress conditions. And it is known in animals that this PARP proteins has wide range of functions ranging from DNA repair to the cell death, depending on the severity of the stress. But we don't know what happens in the plants. So, in my lab, previously it is identified that when we have a more NAD in the plants, it survives in abiotic stress conditions. And also in other study, it shows that when we inhibit this PARP activity, it also shows better resistance to high light. So here we wondered what happens and what is the mechanisms behind this NAD dependent and PARP functions in high light stress conditions. So it comes with two sub-questions. The first one is what are the molecular and physiological responses of NAD-dependent PARP functions in high light, and the second one is if PARP is involved in chromatin modification, and if it is, how does it do that? So why we are interested in highlight? Because in highlight, this is the main inducer of PARP proteins, and also it has a drastic effect on crop yields. So it needs to be resolved to have sufficient food for our future population. So to address my questions, I have set up a condition for highlight and in a combination with PARP inhibitor to inhibit all the PARP and PARP-like proteins in wild type and NAD-enriched Arabidopsis plants. And then to find out my first questions, that what is the molecular and physiological responses, I'm using several techniques. For example, I'm measuring the photosynthetic parameters, and also I'm looking at the expression of NAD synthesis genes and the NAD levels by RT-qPCR and spectrometry, and to see what happens in the metabolic level under high light stress condition, I'm using the gas chromatography mass spectrometry. And finally, to answer these first questions, I also look at the proteins which are pylated by the PARP, and the technique I am using, the LCMS or liquid chromatography mass spectrometry. And to see what happens in a chromatin level, I am visualizing the chromatin modification here by the confocal microscopy, and also to see which proteins are get pylated by the PARP proteins. I am using the confocal microscopy again by and also the immunofluorescence followed by the Western blot techniques. And finally, the last techniques I'm going to use is ELISA. With this ELISA, we can have an overview of what are the other histone modification happened by these PARP proteins. So by doing all of this experiment, we will have the very first picture of this NAD-dependent PARP functions in high light stress condition in plants. So as a perspective, I will continue my research if possible to find and validate the key genes through networking, and ultimately I want to transfer this knowledge from model plant Arabidopsis to the crops, as I want to improve the crops in high light stress conditions. And I would like to thank my team, Team Metabo Action, to help me to conduct this internship, and thank you so much for listening me.
Next speaker is Pei Su. Yes. Okay, let me. Okay. So here the pointer and the arrows. Do you want to try if it works? Yes, it's working. Can you go close to the mic, please?
Hello everyone, my name is Pei, and currently I'm a Master 2 student in Paris-Saclay. And this work is my M2 internship, which is carried out in the lab of PIM, and the school is Ensam. And this work is also cooperate with a PhD student, and her name is Caroline, and my supervisor is called Rustam. The title of my subject is called Numerical Modeling of the Mechanic Behavior of Paper. As we all know, the paper is widely used in our daily life, but they also have the specific function. For example, they can use for packaging and also for the long-term conservation like this. And they can also anti-radiation. And normally when we do the experiment, we can control the, we can put the different tension on the paper, for example, the elastic, and we can see when it broke up. And we can also change the parameters about the storage information, for example, the humidity and also the pH value of the environment, and they also change a lot. And we can impose the different conditions to change the priority of the paper, but how it works, we can see the paper is get damaged, but how they get damaged. And if we want to know the microstructure phenomenon, we have to see smaller, we have to see deeper, and to see the microstructure. So we take a photo of the paper, and we get the SEM photo. And we can see here is the fibers. So we want to know what happened when the paper was destroyed, and in the microstructure. Maybe there have two possibilities. The first one is the fibers, they have been broken, and the second one is the fibers, they are separate. So then we make a model for the fibers. And the numerical modeling, it's actually they have three very important parts. The first part is the model. For example, we can you can also regard it as like the geometry, and there's a lot of nodes and a lot of points, and they also have the line between the node and and you can regard it as like the displacement. And another part is yes, here is a photo for the node and the line, and the second part is the physics priority, because we want to test the we have to compare the modeling and the experiment with the priorities, and they all function on the point and also function on the displacement. And also how we can link them together, we have to find the relationship between the geometry and the priorities. Here we have several methods that are developed well. For example, the finite element method, like the FEM, and also the molecular dynamic, and we also have the other two, and etc. And among all of these methods, the FEM method is widely used and developed well. And to adapt this method, we have the RVE model. RVE model is called the representative volume element, and also we can use finite element analyze to do the post-treatment to get the data and to compare the priorities between the experiment and the model. And then, so what is the finite element method? You can imagine we have the continuous material, and we divided the continuous region to the discrete part, and then we do the approximate in the several parts, and then we organize them together, and you can get a big one. And then there is a 1D model, 1D example for the FEM, and this one is a 3D one. We use we can get a model, and we separate it to the small pieces, and we calculate. We apply the function, apply the different formula in the small pieces, and yeah, there is a lot of step and calculate with a lot of formula with and the metrics. So and another one is a small piece model, and it's called the RVE model, and we have to find the adapter region for the RVE model, because we want to get the large enough model to represent the big one, and we have to get the smallest to cost the computational cost. So we have to find the region, and yeah, the most adaptive method for the human being is to use computer software, and we can also use like the Puma to generate the fibers, and use the parameters from the experiment, and we can also use the Gmesh to do the discrete element, and then use the analyze method, analyze software to calculate the priorities, and then we call back to the model and to check its work or not, and circle. And finally, is another perspective. Yeah, so we can use a lot. It can be used in a lot of fibers, and we can also use the multi-scale, the different modeling to do this. And maybe we can also apply the machine learning and AI to for this numerical modeling work. Okay, so that's all my presentation. Thank you for your attention.
Thanks. So our next speaker, Helen. Ellen is here? Yeah? Okay. Okay, can I try to be close to the microphone? So this is the pointer and this is the arrows to the place.
Ladies and gentlemen, good evening. My name is Ellen JC, and I'm an M2 intern at the Université Paris-Saclay, Institut de Chimie Physique. So under the supervision of Prof. Chantal Houé-Levin and Dr. Laura Basio, I investigated a topic, flash irradiation, beneficial or not for non-specific immunity. Basically, we want to know if flash irradiation has a beneficial impact on our immune cells. So over the years, radiotherapy has been used in the treatment of cancers. It uses ionizing radiations to target and kill cancer cells. Unfortunately, this leads to unintended consequences such as compromised immune system, lethargy, and even hair loss. There is a newer method, that is flash irradiation, that has proven to be beneficial to healthy cells as compared to the conventional radiotherapy. Let us look at the differences between both of them. So for the conventional radiotherapy, that is what we have right now in most of our hospitals. It uses low dose rates radiations and it spreads radiation doses over multiple sessions. While with the flash radiation, it gives a high dose rate of radiation in just a few or a single session. For the biological effects, the conventional radiotherapy affects cancerous cells as well as healthy cells. But for flash radiotherapy, it has been proven through computational studies and experimental models that it affects just the cancerous cells. Some advantages of the flash radiation is that it will reduce the treatment time, it will spare healthy tissues, and it will increase patient comfort. Now, we know that this new method is promising, but little is known about the mechanisms that are involved in them. This research aims to basically know the effect of, or to characterize the effect of flash radiation on our immune cells with specificity to our neutrophils. So at the cellular level, we wanted to examine the lifespan of neutrophils as a function of dose. So when you give irradiation to neutrophil cells, that is purified neutrophils, we want to see how fast they die after giving them radiation. We also wanted to check the cell's ability to undergo a specialized form of cell death by neutrophils known as netosis. At the molecular level, we want to examine the state of NADPH oxidase enzyme and also examine the effects of irradiation on the rate of production of superoxide. For this experiment and what I'll be talking about, the molecular level has not been yet experimented on. So this is our first diagram. This is a graph showing you the cell survival after we gave them radiotherapy for conventional and flash. The conventional radiotherapy uses gamma or X-rays, while the flash uses high-energy electrons. So for the gamma irradiation, we isolated neutrophils and gave them different doses of radiation. After that, these are the doses. Our sham means that it did not receive any radiation. 2 gray and 15 gray are middle doses of radiation, and 47.9 gray is considered a high dose of radiation. So after that, we measured how fast the cells die after giving them the radiation. After the 4th hour, we observed that the highest dose had undergone almost 100% of cell death, while the middle doses had undergone about 60% of cell death. Our cells, which did not receive any radiation at all, underwent about 40% of cell death. Also, the experiment was repeated with flash radiation, and it was observed that at the 5th hour, only 50% of the cells were dead at the given high doses, that's the 25 and 50 gray. And the lower doses and the sample that did not receive radiation at all was about 15% of cell death alone. We also investigated if the cells were able to undergo the specialized form of cell death, that is netosis. We've performed the experiments with gamma radiation and we've observed that it undergoes netosis, but we've not been able to yet experiment on flash radiation. So in conclusion, we observe that the cells undergo cell death much faster with gamma irradiation as compared to flash radiation. Thank you very much.
So our next speaker is Amit. He's not here. Well, is Maxime Coca here? No, I mean neither. So, well, I just want to take the opportunity to thank for excellent presentations you have done. Very nice, and that showed actually the great diversity of our university. So it's really a pleasure to be with you and to discover your work. And I think that you all deserve an applause. Thanks a lot. And now we go back to the hall. Okay.
Dommage, la deuxième vidéo là, sur la théorie des groupes.
Non, de toute façon, les animations étaient super.
Il parlait trop vite et puis le son était pas.
En fait, il a laissé allumer le micro, comme ça faisait l'écho. Et oui, aussi.
En plus, la présentation avait l'air super bien faite.
Sauf que la première scène, par contre. Les animations, bah, la vie du même monde, le mec qui prend l'inspiration des meilleurs, ça me fait plus de
Mais bon, niveau auditif, c'était pas du triple one road.
Voilà, le truc, qu'est-ce qu'on a fait, tu peux voter pour toi-même. Si, tu peux voter même, n'est-ce pas? Tu peux voter pour toi-même.
Ah ouais, mais c'est quoi?
Non, c'est pas ça. C'est la vie, c'est la règle du jeu. C'est le coup du public, donc toi tu peux voter qu'une seule fois, moi je peux voter qu'une seule fois. Tu auras 2 votes pour toi-même. Je veux dire. Il faut que tu ramasses encore une cinquantaine pour gagner quelque chose, t'es d'accord, non? Donc lui, il peut te voter pour toi aussi, ça fait 3.
C'est vrai.
Mais après, un nombre limité de potes...
C'est vrai qu'après, s'il avait rempli la salle de 6 stars...
Alors la vidéo, qui est tout à fait...
C'est suffisant pour être admissible, mais c'est vraiment pas assez. J'ai 14 000.
14 Denis, c'est pas assez.
moyenne. Bah, derrière, il y a les euros. Je pars pas avec une bonne avance, quoi.
D'accord. C'est combien une bonne note de ta timicile centrale?
C'est vraiment en termes de points, je sais pas trop différent. La barre est à nous deux.
Là, la barre, elle est à 13,5 sur 20. J'ai à peine un point de plus que la barre des admissibles.
Et
c'est pas...
Est-ce qu'il y a 800, presque 900 personnes devant toi?
Victor Minot, il était à 0,5 points de plus sur 1000 pour être admissible à Chipote, il y allait à Chipote.
Non, regarde quelque chose comme ça. On admet, on parle des chiffres qui ont été passés, 800 élèves. Les 2 dernières années, 800 et quelques. C'est pas 900.
On va
c'est plus que 800.
plusieurs filières aussi.
Plusieurs filières, d'accord, non, mais. Non, c'est pas plusieurs filières. C'est par contre généraliste. Est-ce qu'il y a 1000 personnes devant toi? Si ce n'est pas le cas, et que tu es admissible, tu veux bien aller accepter.
Je regardais justement.
Comme il y a les gens qui passent dans le concours, par exemple, de Leeds, ils peut-être vont préférer aller à Leeds, parce qu'ils ne veulent pas faire une prêt pour les ingénieurs.
Ouais, l'année dernière, il y avait
Peut-être ils vont à Leeds, et voilà.
Oui, bien sûr que ça va libérer des places. Après, l'année dernière, il y avait 600 admissibles et sur la CEI, ils ne donnent pas le rang du dernier ami, je sais pas pourquoi. Mais
La médiane était à combien?
la médiane était à 200. Non, mais ça se fait. Ça se fait. Mais bon, faut pas que j'ai pas le droit à une semaine, hein, je pense. Et là, à côté, il débarque 17,8 de moyenne sérieuse. Un ami qui fait 5 de milieu aussi. 17,8 de moyenne.
Après,
je comprends pas parce qu'en plus, entre guillemets, pendant tous les délais de l'année, j'étais devant lui pour en sciences.
Moi, ce que je ne dis pas que j'entends de ça, c'est pas le fait que vous rentrez ou que vous ne rentrez pas. C'est comment vous, qui rentrez avec par exemple 17,5 ou 15, vous arrivez peut-être en physique quantique ou au DP ou en cours de première année, vous avez du mal à avoir 10.
Alors, il y a un autre parallèle qui rentre dans l'équation.
Ouais.
Mais ouais, en plus,
ouais, je suis pas encore intégré donc je peux pas me prononcer.
Ok, je vais te dire une chose qui reste et toutes tes profs de prépa t'ont dit. En fait, ta progression dans le milieu de l'enseignement supérieur, elle a commencé pendant le prépa. Mais elle ne se finit qu'au moment où tu sors de la supérieure. C'est-à-dire pas plus seul, dont 5 ans de travail. Certes, tu as beaucoup bossé pendant les 2 premières années en prépa, mais prendre des vacances pendant 3 ans, ça fait exactement ce que je viens de dire. Tu auras du mal à voir 10. Et tu sais que c'est ça une partie du problème. On a beaucoup de touristes dans l'école. De vacanciers. Ouais. C'était pas faire ça.
Statistique et apprentissage, j'ai révisé 5 jours avant, sérieusement, et
Et pourquoi physique quantique, t'as eu du mal? Moi, je me rappelle que tu as.
Bah, en fait, c'est
T'as cassé, hein?
Je
ne sais pas quelle est sa note, mais je sais qu'il a pas mal bossé.
Bah, sinon, du coup, ça c'est bien marché.
Et Théo, c'est un bon élève, il n'y a pas de problème. Par contre, il y a un tiers de la promo. Je sais pas qu'est-ce qu'ils font là. Hein.
Sérieux? À ce point? Ouais, bon, quand il y a 2 soirées par semaine.
Oh, oui, mais si je te dis simplement qu'il y a TP, d'ELEC ou de l'électrotech noté et que cela vaut 20% de ta moyenne, tu fais quoi ce soir?
Non, je fais
tu prépares le.
De toute façon,
c'est cool de ne pas le préparer. Tu veux noter quelque chose, préparer les formules. J'en sais rien.
Les réponses sont notées sur le tableau, il faut juste les copier.
Oui. C'est pour ça que je dis que tu ne fais pas ton TP. Ça veut dire que tu ramasses toutes les choses et tu mets en ordre. Tu dis tu viens dans le TP avec les trucs en ordre. C'est juste ça qu'il faut faire. Ça prend 10 minutes.
Il y a un tiers des élèves qui ne le font pas.
Oui.
Ah, pourtant, je suis convaincu que associatif et modèle, c'est pas du tout
Non, tu peux faire de l'associatif. L'associatif, ce n'est pas un problème. J'ai été président d'une association pendant 2 ans dans mon école d'ingénierie alors. C'était
franchit pas de